Hi, my name is Yolande. I'm from Towson University, the Department of Biology, and I will be presenting the determining optimum pH for trypsin enzyme. So, an enzyme is a substance produced by a living organism that acts as a catalyst to bring about a specific biochemical reaction. Enzymes can be proteins or catalysts that breaks down reactions by lowering the activation energy without taking part during the reaction. And they can be catalysts both and then affect the reaction rates of the and then the difference between catalyst and enzyme is that while catalysts are inorganic compounds, enzymes are largely organic in nature and are biocatalysts. In living organisms, enzymes speed up reactions and regulate reaction rates. Trypsin is a proteolytic enzyme that is synthesized in the pancreas in its inactivated form, trypsinogen. And as a protein, trypsin cleaves after ARG and TYR, so it is a hydrolysis protein of peptide bonds. Trypsinogen is then converted into the form trypsins by the small intestine where its interest is to facilitate the digestion in the body according to Fortingen. So trienzyme breaks down proteins by breaking down the peptide bonds into smaller peptide chains, where the trypsin is one of them, which is activated at a pH of 8 and is produced in animals by small intestine, while inactivates found in a slightly acidic environment in the pancreas where the pH is around 7. However, bacteria also secrete trypsin in the environment at the pH ranging from 7 to 10, according to Pilon. So the group decided to focus on the pH condition that trypsin may experience and more specifically finding the optimist pH enzymes activity at around 9. So for the purpose of this experiment, we hypothesized that pH of 9 would be optimum for trypsin and the most reaction will occur at that pH. So prior to that, the materials we used were the reaction buffer, the azocasine, we had trypsin of 2 milligrams and the azocasine was 0.5%, and we also had pH of a kept on ice. For the experimental control, we used the DI water and we had 10% of trichloroacetic acid or TCA solution, and we had the test tubes, the temperature controlled dry bath at 37 degrees Celsius. We had the test tubes, the centrifuges, the cuvettes, spectrophotometer, pipettes with the serological pipette, transfer pipettes, and micropipettes as well. So prior to that, we decided to make 4 piquets of the experimental and the negative control for each pH that were tested. So in total, there were 20 tubes of the negative controls and 20 tubes of the experimental controls for a total of 40 that we all labeled to avoid confusion. So we used a serological pipette to pipette about 5 to 10 milliliters of azocasine and 0.15 moles of sodium with 20 ml twice ACL with pH of 7, 8, 9, and then the 0.15 sodium of 20 ml borate for pH of 10 and 11. So they were then placed into different tubes as labeled. In each of the centrifuge tubes, we used the 1000 microliter pipette to add 450 microliter of the reaction buffer for the 4 piquets for each pH, and we also added 500 microliters of azocasine. Then we used the 200 microliter pipette to add 50 microliter of DC water for the negative control and 50 microliter for the trypsin for the experimentals. So the test tubes were then closed and mixed by inverting. Then we started the time as soon as we put the enzyme prior for incubation. For the incubation, we place all the microcentrifuge tube inside of the dry bath at a temperature of 37 degrees Celsius for 20 minutes. And after we incubate all the centrifuge tubes, we put them gloves then before handing the TCA. So for the TCA, we added 500 microliters with the 1000 microliter pipette of the 10% of the TCA to inside of each our 40 centrifuge tubes, and we mix all the solutions after we close them. Then we centrifuge the tube at a maximum speed of 12,000 RPM for 2 minutes. Then the centrifuge capacity, which is 18 tubes, we had to make 3 centrifugerations for our 40 tubes. After that, we label all the cuvettes. So we had a total of 40 cuvettes at the same weight as we label all the centrifuge, we label all these cuvettes for the spectrophotometers. So we turn on the spectrophotometers, we put the spectrophotometers at 440 nanometers, and we blank the spectrophotometers with a cuvette filled with DI water. So after that, we read the absorbance of all our negative controls and all our experimental controls too. And after that, we had all our results and we compared the results between both samples. So the TCA was then added to stop reaction. TCA is a catalyst and it can also be used to break down reactions as well. So it was also used there to precipitate the undigested substrate. The solution was centrifuged after adding TCA because TCA precipitates enzyme to catalyze reactions by removing undigested substrates. After we centrifuge the tube, sediments from the reactions is then at the bottom of the test tubes, and then we had the supernatant at the top of the test tube as well. And these are all our results. So we had the substance used for the enzyme that was 10% of azocasein, and the reaction buffer for HPH used. So the addition of sulfonamide is correlated to the absorbance of 440 nanometer because we chose the temperature to be 37 degree because body temperature is usually at 37 degree, but then for the 440 nanometers for the spectrophotometer was because we added a milk protein, which is the trypsin, that absorbs light at 440 nanometers. After that, we assumed the pH to be 9 because trypsin reacts in the small intestine at the pH close to 9. So there were equal numbers of negative controls, and we also decided to make the same amount of number for the experimental controls because more replicates is good for more accuracy of the solution. And also we had 50 microliters of trypsin to the solution, and we added 50 microliters for our negative, while where the azocasein had the GI water in it. So we stated that since it had GI water, the negative control with the GI water wouldn't affect the reaction at all. So these are our results. Here is the enzyme activity and the pH level. The highest ones for the values are the experimentals, and then the controls are the lower ones, and then in between we added the standard deviation. Is much higher on the experimental controls than on the negative control right here. So on average, there is about 21% difference between the average absorbance values for the experimental group versus the negative control group. And here we had the result with our pH and the t-test and the pH at 9, and also the t-test control. So for our t-test control, all the results were less than 0.05, whereas for the t-test and the pH at 9, we had almost all of them were greater than 0.05, except for the one that pH of 11 that had a reading t-test versus pH 9 less than 0.05. So prior to that, we stated that the enzyme had more activity at pH of 11, which does not support our hypothesis. So the t-test is usually calculated to determine a significance difference between negative control and experimental. So the t-test between the experimental trials suggests that there may have been too much enzyme present in the samples or other errors. So the t-test between the experimental and the control group suggests that there is some enzyme activity in the experimental as well. But it was hypothesized that enzyme activity would be higher at a pH of 9. So the results does not support our hypothesis, and there was no significant difference between the absorbance reading of any of our experimental group. Even for our negative control, the absorbances reading were very close, and we can see that on this chart right here. So after the after making that result and the differences, we compare our result with the other groups, and then we found that our p-value, but first of all, the p-value, which is a result of a t-test, so showed variability within each experimental group compared to the variability of the p-value within each experimental group compared to the variability between sample sets. A p-value of 0.05 or less, usually, in order to say the independence variable is the factor responsible for the variation, while the t-test done for pH 11 had a p-value lower than 0.05, and other t-test value are greater than 0.05, so therefore they were all inconclusive. So the choice of control variable for our experiment after seeing the results of the other groups stated that for the temperature we could have improved it, because the per group found that the optimum temperature for a maximum enzyme activity was between 48 to 68 degrees Celsius, and we only had 37 degrees Celsius, so we should have added the temperature, increase it, or probably had added more replicates maybe to our experiment to make it more precise, or maybe we could have blanketed the spectrophotometer wrong. That's why we had like bad results, not like good result on our readings, or maybe we left our test tube too much inside of the dry bath, or maybe we could have pipetted wrong because our test tube had like an equivalence number of microliters in it for all our centrifuge tubes. So, as trypsin is used as a digestive enzyme by bacteria, using the result from each experiment, the ideal condition for growing bacteria can be found. As outliers, before the ideal temperature for maximum enzyme was found between 48 and 68 degrees Celsius, the love group, however, proved that adequate to conclusively say the optimal pH level for maximum enzyme activity will be best between pH 7 to pH 9. So, after those possible errors, we decided that they could have sources of variability and differences the pipetting technique or leftover of the supernatant, or we could have like some of the pellets inside of our cuvettes. And here is our work cited. Thank you for watching.